Merci, merci Marie-Chantal. C'est vraiment un très grand plaisir d'accueillir Dr. Lakkis. Ça fait 8 ans, la dernière fois, je pense qu'il est venu au CRCHUM. On venait juste d'ouvrir le CRCHUM. J'étais très enceinte de 41 semaines. On venait de déménager le labo. Et puis, Dr. Lakkis a visité. I'm just saying that you visited 8 years ago. And since then, things, a lot of science went on, and I'm really happy to have you here. So, juste pour expliquer un peu, Dr. Lakkis est professeur émérite de chirurgie et professeur d'immunologie et de médecine à l'Université de Pittsburgh. Il a obtenu son diplôme de médecine à l'Université américaine de Beyrouth au Liban et a suivi une formation complémentaire en néphrologie et immunologie de la transplantation à l'hôpital Beth Israel à Harvard Medical School. Après avoir fait partie de la faculté de l'Université Emory et de l'Université Yale, Dr. Lakkis a rejoint l'Université de Pittsburgh en 2005 en tant que titulaire de la chaire Frank Tinastaris en biologie de la transplantation. Et il est directeur scientifique de l'Institut de transplantation Thomas Start, financé par le NIH. Les travaux du Dr. Lakkis portent sur 2 domaines fondamentaux d'immunologie de la transplantation, soit l'alloreconnaissance dans le système immunitaire inné en utilisant des modèles de transplantation chez les invertébrés et les vertébrés, puis la biologie des cellules T mémoire dans la transplantation d'organes chez les animaux et les humains. Dr. Lakkis a reçu de nombreux prix et reconnaissances. Il a reçu le Young Investigator Award de l'ASN en 2002, l'American Society of Transplantation Basic Science Established Investigators en 2009. Il est membre élu de l'American Society of Clinical Investigation et de l'American Association of Physicians. Et le Dr. Lakkis a également siégé au conseil d'administration de l'American Society of Transplantation. All that to say that it's a real pleasure to have you here, Fadi, and you published recently really amazing and fascinating observation on innate immune memory. I was saying when I did my immunology classes, if I would have mentioned innate immune memory, people would have called me crazy, but I'm really glad that you're here to explain to us your observation. Thank you.
Thank you very much, Melanie and Marie-Chantal, for inviting me. And I wish I were there in person. It's always a pleasure to go to beautiful Montreal. So the title of my lecture is both innate allorecognition and innate immune memory, because it would be impossible to explain the memory without first explaining the innate allorecognition phenomenon. So what I would like to do is first start with what I think is a central paradigm in immunology, which has stood the test of time, which is the paradigm of self and not self. This paradigm was advanced by Sir Macfarlane Burnet, who went on to win the Nobel Prize for the clonal selection theory and for immune tolerance alongside Sir Peter Medawar. So as you will be able to tell by the end of my lecture, the story that I will tell you is not only about self and not self, but also some form of clonal expansion and clonal selection in an unexpected cell type, which is the monocyte. So when we talk about non-self in immunology, we can think of 2 big baskets. One is microbial non-self, which are the pathogens, and that's what most immunologists study. But we are transplanters, and we are very proud of that. So we also think of allogeneic non-self. And I think it becomes in at least 3 different flavors. One is the kidney or heart or lung transplant that a surgeon puts in. But also fetuses are allografts, and the mother has to deal with that. And one can even think of tumors, especially ones that have a high mutational burden, as allografts that have arisen from within, from inside the body, because now they look like a tissue that has been transplanted if they have many proteins that are different from that of the host. So how is it then that the immune system senses non-self in general? If I'm telling you that the immune system is all about sensing non-self, and that's how it knows whether to respond or not. How does it do it? Of course, we all know how the adaptive immune system, meaning the T lymphocytes and the B lymphocytes, are able to recognize non-self. They do that using these very advanced receptors, the T-cell receptors and the antibodies that arise by a somatic gene rearrangement. They're highly diversified, and they're able to detect the slightest differences between the host and the pathogen, or the host and the grafted tissue. So they have very high molecular specificity. But as all of you know, it's not enough to activate the adaptive system, or to say it more correctly, the innate immune system has to be activated before the adaptive system can be activated, because of the reliance on dendritic cells or antigen-presenting cells that have to present the antigen in the context of MHC molecules to these very fancy T-cell receptors. And the antibodies may not require dendritic cells, but they also, in many ways, require innate immune activations. So it wasn't until Charlie Janeway back in the 1980s that we then understood that even the innate immune system, made up of, for example, myeloid cells, is able to recognize non-self. And this non-self that was discovered then were patterns or common motifs that are present in pathogens. For example, bacteria, parasites, fungi, and viruses that are detected by pattern recognition receptors, or PRR, on the surface, for example, of macrophages or dendritic cells. And all of you know the story of the toll-like receptors and how this field of innate immunity has mushroomed and has led to improvements, for example, in adjuvants for vaccination. The question that we wanted to ask, and believe it or not, it's been almost 15, 20 years now, is how is it that the innate immune system can sense an allograft? And does it sense it in a way that is specific, just like we sense viruses and bacteria? Or is it just a random event, for example, danger, where you have inflammation and that's all it takes? Or is there some kind of receptor ligand system that tells the innate immune system, specifically the dendritic cells or myeloid cells, that they're really looking at something that's not them, that is foreign, and therefore they must react? So the way we decided to address that was, of course, in the mouse, because it's simpler, and you can use reductionist models. And the model that we used is the RAG knockout gamma chain knockout mouse, which completely lacks all lymphoid cells. So it lacks NK cells, lacks T cells and B cells. So you can think of it as an innate mouse. So we placed either an allogeneic or a syngeneic graft into the mouse, and then we compared the immune response. In this case, is a completely innate response. And we hypothesized that if there is no difference between an allogeneic graft and a syngeneic graft in terms of the response, then it must be that the innate immune system does not sense non-self. It just senses inflammation, the inflammation associated with the surgical procedure, for example, and it doesn't care where the tissue came from. But if we did find a difference, that would be our first step or insight into some kind of sensing of non-self that we can then explore further. So we did the experiment, and we did find that there was a difference, and the difference was in what we refer to as the monocyte-derived dendritic cells. So it turns out that when you transplant an organ, there's an influx of monocytes from the recipient, and these monocytes change into dendritic cells. Some of them change into macrophages as well, but here I'm going to focus on the dendritic cells because that's where we found the most pronounced difference. And the difference is shown here. If you put a syngeneic graft and you look at these solid circles, there is a rise in the number of these mature monocyte-derived dendritic cells. These are dendritic cells that have costimulatory receptors on them, costimulatory molecules, I apologize. But the number of these dendritic cells declines very rapidly and becomes negligible over time. In contrast, if you put an allogeneic graft, you realize that there is a larger number of these dendritic cells. They do come down in number, but they persist over time. And keep in mind that since these are gamma chain knockout rag knockout mice, they do not reject the transplant. You still need a T cell to reject a transplant, but they allow you to study the transplant over a long period of time. So the point is that if you put an allogeneic graft, it's not the same as putting a syngeneic graft into the animal. And the difference is in the number of monocyte-derived dendritic cells, which are mature. And they, in fact, can also make interleukin 12, which drives the canonical Th1 or interferon gamma response that we very much associate with rejection. So what I'm telling you is that there is an event where the innate system is sensing non-self, and that sensing is driving the maturation of dendritic cells that we know very well can drive T cells that can cause rejection. So that, in our mind, was the first link between an innate recognition event in the context of transplantation and the adaptive immune system. So is this link really important? We think it is, and we've worked on that to test the hypothesis that these monocyte-derived dendritic cells are indeed important for rejection. The first step was to ask how much they are, or how many of them there are. And as many of you know, when you put an organ, a transplant, like a kidney or a heart or a lung, into a human being or into a mouse, it arrives with a large number of dendritic cells that reside in the tissue and come from the donor. They are shown here in yellow in this kidney mouse transplant experiment. And however, these yellow cells start to vanish, and they are reduced by more than 90% within a few days. And by 7 days, they are still there, but they're in a minority. And instead, you see this massive infiltration of the kidney with recipient-derived dendritic cells. And we could determine that they were of the monocyte variety. They're CD11b positive, CD11c positive, for example, and they take over. So what is it that they're doing? And it turns out that they are important. They are important for at least two things. One, they help with the migration of T cells. So T cell migration into the graft turns out to be antigen dependent. And this is a paper that we published in JCI a while ago. And I'll just show you this movie of a green host-derived dendritic cell that has captured the T cell, which is in red, inside the blood vessel. So these dendritic cells extend dendrites into the lumen of the capillary, and they can drag the T cell out. And if you play tricks so that the dendritic cells cannot present antigen, or if the T cell cannot see the antigen, the T cell doesn't migrate. So this initial step of T cell migration into the graft is antigen dependent. It's dendritic cell dependent. And of course, then there are chemokines that produce, that bring in additional cells. But it's all dependent on this antigen recognition step. Again, goes back to the idea that the immune system is driven by this self, non-self recognition, and not just by non-specific inflammatory phenomena. It also turns out that these dendritic cells are important for propagating the immune response within the organ. So we all know that immune responses, naive immune responses, are initiated within lymphoid tissues, such as lymph nodes and the spleen. But once they're initiated and you have effector or memory cells, these cells can then migrate to the tissue, and that's where they can sustain themselves and even form, for example, resident memory cells. So we asked, what is the role of these monocyte-derived dendritic cells? And we could clearly show that if you were to deplete them, they're CD11b positive. This is a short-term experiment because the depletion of CD11b positive cells can be quite toxic to the mouse. But even within 7 days, you could see not only that we've cleared, not shown here, that we've cleared the dendritic cells, but you also start clearing the T cells. And if you do a long-term experiment using a CD11c depletion model, you can show that by depleting, in this case, it's a CD11c DTR. I don't have to get into the details. You administer DT and you deplete the DCs. You can show that you can prolong allograft survival, even if you come in with the DT as late as 5 to 10 days after transplantation. So what this told us is that monocyte or dendritic cell depletion, even after rejection has been initiated, can slow down rejection over time. proving to us that these dendritic cells are playing an important role in the graft. I'm not saying they're the only shakers and movers, but they're without doubt they are important. So now we then get really to the essential question, or the most fundamental question. What is it that these monocytes are sensing? We know what T cells sense. We know how they see antigen in the context of MHC molecules. We know what B cells can see. But what is it that monocytes are sensing that is non-self on the organ that's leading them to mature into dendritic cells, and then those in turn can activate T cells or can sustain the T cell response within the organ? So the story I'm going to tell you today is largely focused on monocytes, but much of what I will tell you also happens in the transition from a monocyte to a macrophage, and I will acknowledge people at the right time and at the right place. But for the sake of time, I'm just going to focus mostly on the monocyte story, because I think it's sufficient to make the point of what they are sensing. And then I would move on to the concept of memory in monocytes and what the molecular mechanisms are. So in order to know what they're sensing, we decided to do initially a sort of an unbiased experiment, or a genetic screen. So what we did, we kept the recipient constant, a B6 RAG knockout gamma chain knockout mouse. It doesn't have an adaptive immune system. It only has myeloid cells. So whatever we detect in it must be mediated by the myeloid cells. And we varied the donor. And then we used, again, this number of monocyte-derived dendritic cells as the measure of the activation of the innate immune system in response to allogeneic non-self. So we used a wide variety of mice, and we could see that there was a variety in the response. And the biggest difference we could see, or the most interesting difference, was between the NOD donor and the NOR donor. So NOD and NOR mice are highly related. They're about 90% identical in their genomes. And nevertheless, one induces a very strong innate alloimmune response, while the other induces a very weak one. And in fact, if you do the experiment in the same mouse, we played a trick here. Instead of putting a kidney transplant or a skin transplant, we put a bone marrow plug under the kidney capsule, so that you can put one under each capsule, under each kidney. You can come back and analyze the number of cells that have infiltrated. And you can see that really the NOR behaves like a syngeneic transplant, while the NOD is significantly greater than either the syngeneic transplant or the NOR transplant. So now you have two donors that are genetically 90% identical, yet they elicit strikingly different responses. You can do a genetic mapping study if you had congenic mice that narrow the genetic segments gradually. And we were lucky to collaborate with an investigator at the University of Toronto, Jane Daska. And we were able, using this genetic mapping strategy, to identify SERP-alpha as the polymorphic molecule on the donor that's being recognized by its receptor CD47. So let me summarize a whole bunch of work in one slide to tell you how this system works. So this is an MHC independent system. This has nothing to do with the MHC. This is a polymorphic molecule that's eliciting an innate immune response that is not an MHC molecule. So the way this works, it turns out, and I'll show you the experimental proof for that, is that when a monocyte encounters self, it is encountering a SERP-alpha molecule on a self cell that is recognized by CD47, which, by the way, is not polymorphic. And this can actually lead to a stimulatory signal. However, as many of you know, and your groups have done beautiful work on SERP-alpha, SERP-alpha is an inhibitory molecule. It's a do not eat me signal that is present on monocytes, macrophages, and dendritic cells. So it tells the monocyte not to mature. It tells the macrophage not to eat the target cell. So when the SERP-alpha molecule on the presenting cell as well as on the responding cell are identical, the signals are equal in intensity, and therefore no response ensues. However, if you now put in a transplant and the non-self cell has a different SIRP-alpha, particularly if that SIRP-alpha tends to have higher affinity to the ligand CD47, you create an imbalance in the signals, and that causes the monocyte to mature into a dendritic cell. You can test this, and we did it in two ways. You can specifically block the interaction between the non-self SIRP-alpha and CD47, and you can show that you can completely abrogate this monocyte to dendritic cell maturation response. Or you can use a recipient that lacks CD47. So the donor has CD47. It can signal this way, but cannot signal that way. And that then leads to, again, abrogation or prevention of this innate alloimmune response. So it looks like it's a novel system, although SIRP-alpha itself is not a novel molecule, working in a funny way because the molecules are on both sides, on the responder and the donor. However, it makes sense, and you can test it experimentally. So it doesn't mean anything in the context of transplantation. So we did test that, and this is the work of Dachen Zhao that we're getting ready to be published, because as I will show you, we can take it all the way from mice to humans, where we show that in the absence of the CD47 molecule, or as I will show you in a minute, if we manipulate SIRP-alpha on the donor, we can ameliorate chronic rejection. We do not affect acute rejection. We only affect chronic rejection. So this is a kidney transplant model where the donor is a nod, while the recipient is a B6 background that's either wild type or lacks the receptor for SIRP-alpha, which is CD47. And you can see that we significantly reduce the infiltration in the kidney as well as the fibrosis in the kidney. And in the mouse, kidney transplants are not rejected acutely, they're rejected slowly. So they serve as a quasi-chronic rejection model. Then we can do a different experiment where we can vary SIRP-alpha. So we keep the recipient the same. It's a wild type B6, but we either put a nod donor or a nod donor that's congenic for the B6 SIRP-alpha, meaning that it is MHC different from the recipient, but we were able to make the SIRP-alpha the same as the recipient. So we took away the SIRP-alpha mismatch, and we can show again that the amount of infiltration and the amount of fibrosis are now reduced, and it becomes more similar to a CD47 knockout recipient. So this suggests that by manipulating one polymorphic non-MHC molecule on the donor, we are able to alter chronic outcome in a graft. So what about humans? So it turns out that we as humans have several haplotypes of SIRP-alpha. They're not, of course, as variable as the MHC, where you have 10,000 or so altogether. Instead, we have about 10 or 11 haplotypes that can account for 99% of the variability in humans. And even more so, one or two haplotypes account for the majority of what you see in humans. So for example, haplotype number 1 has the highest frequency, followed by haplotype number 2 here. And there's some variation based on the ethnic background of the individual. So for example, haplotype number 2 becomes more common in East Asians, while haplotype 1 declines in prevalence compared to other ethnic backgrounds. And it turns out that you can actually do a nice PCR reaction that could divide these most common haplotypes into two groups, one with an amino acid deletion, the 3 nucleotide deletion, and one without it. We call them A and B. So now you can do studies on that. I'm not going to put you through the torture of telling you all the studies. I'm just going to show you one piece of human data. And we're expanding on it, where we genotyped 86 donor-recipient pairs. They are all living donors. So these are recipients of living donor kidneys. So we genotyped the donors and recipients, and we divided them into either matched or mismatched for SIRP-alpha. And you can see that after the first 12 months, they start separating, and they separate significantly over time, where people with a SIRP-alpha mismatch tend to do a little bit worse over time than those without the mismatch. So this is showing us that there is a locus outside the MHC that can influence graft survival, and we have the biological explanation for that, which is recognition of this molecule as non-self by monocytes. So we're really opening up a new area here where it's not only about T cells and B cells, but it's also about these myeloid cells that can sense non-self and can do damage over time. So let me now move to the subject of memory, which is really what this lecture is supposed to be about, and it's about allospecific memory. So let me just say a couple of words about immunological memory. So if you were to look it up in an English dictionary, let's say the Oxford Dictionary, it will tell you that immune memory is specific to the original encounter. So it's not just an enhanced response after a second exposure to an antigen, but it has to be specific to the first antigen. So it can't be just an enhanced response to just any insult or any antigen. It has to remember what it had seen before and only respond to that first encounter, to that antigen that it had seen in the first encounter with greater cell proliferation or greater immune responsiveness. So, and as you know, some forms of memory have been described in the innate immune system, definitely very clearly in the NK cell system, but not as clearly in the myeloid cell system. People talk about trained immunity, but that is of the non-specific kind. It's angry cells that remain angry for 4 to 8 or 12 weeks, but in reality, they don't remember what they responded. You can re-stimulate them with anything and they'll act angrily to it. So what we did here, we immunized mice, these mice that lack lymphocytes or NK cells, and we came back a week, 4 weeks, or 7 weeks later, and we put in a graft that's either identical to the immunization or different from the immunization, and we asked whether we see an enhanced response, and if we do, is it specific? So what we found is that we do for both questions. In other words, we do see an enhanced response. So in this case, the recipient mouse, the B6 RAG knockout gamma chain knockout, were either left unimmunized, was immunized with syngeneic irradiated spleen cells, or with allogeneic cells, or with third-party cells, C3H. And we came back a week later, 4 weeks or 7 weeks, and we placed a BAUT-C graft. And you can see that there was an enhanced response to the BAUT-C graft only if the mouse had been previously, had been previously exposed to BAUT-C cells, but not if it had been exposed to C3H cells. And we could see this enhanced responsiveness that is specific to the original encounter at 28 days, but it had completely dissipated by 49 days. We could switch things around and now show that if you had immunized the mouse with C3H, you can enhance the response to a C3H graft, while BAUT-C immunization, which had done a great job with BAUT-C grafts, it did not do anything to the C3H graft. It went back to the level of a primary response. So this proved to us specificity, and we even did it in the complete absence of T cells, B cells, and NK cells in both the donor and the recipient. So now the donor graft, as well as the splenocytes that we use to immunize the mouse, come from mice that are on the gamma chain RAG knockout background, and so does the graft. And again, we see this enhanced memory response. In this case, we did it at 7 days. And finally, we could show that, I'm not sure why I'm showing you this. It must have been a control experiment. So I apologize, I'll skip it. But the point is that there is memory, it is specific, and it's not dependent on lymphoid cells, either in the donor or the recipient. So is it really mediated by monocytes? And the best experiment you can do with memory is to show that you can transfer it. That's how T cell memory has been studied in the mouse for many years, and perhaps in humans in some ways when we do adoptive T cell transfer. So what we did here, we isolated monocytes from mice that were either immunized with syngeneic cells or allogeneic cells, and we could show that they can transfer memory, they could transfer this enhanced responsiveness to the graft, even if we harvested them 7 days, 21 days, or 35 days later. So not only showing that the monocyte is the cell mediator of the memory response, but we're also showing longevity, that even if it comes several weeks after the original immunization, these monocytes are able to retain that memory and transfer it to a naive mouse. So what is mediating this memory? What molecules are mediating it? What is it that the monocyte is sensing that is allowing it to become a memory cell? So of course, the first question, being transplanters and showing you the experiments I showed you before, is whether it is dependent on an MHC mismatch. In other words, is the non-self molecule that is recognized an MHC molecule? And the answer is yes. And we know that because we can take away MHC at the time of priming using congenic mice. We show that the memory response goes away. We can take the MHC mismatch at the time of recall. We show that the recall response goes away. And then finally, we show that an MHC mismatch at the time of priming alone is not sufficient. There's something else happening. And at the end of the lecture, I'll tell you it's SERP alpha again. But the point here from this slide is that unlike the SERP alpha story, we're dealing with something, a receptor that's recognizing the MHC. And remember, these are monocytes. They don't have immunoglobulin molecules. They don't have T cell receptors. So what is it that they're recognizing? Before I tell you what the receptor is that's recognizing the MHC, this is just another experiment to show that indeed that it is the MHC1 molecule specifically that's required for this memory. In this case, this happens to be a macrophage experiment, not a monocyte experiment, where you can show that if you immunize a mouse with allogeneic cells, the macrophages gain cytotoxic activity that is specific to those allogeneic cells. So you can see that all the BALB-C cells here disappear in this in vivo cytotoxicity experiment. But now if the donor cells are beta-2 microglobulin deficient, meaning they don't express MHC class 1, you lose much of the cytotoxicity. You can see them again. Or if you use a congenic animal where there are minor antigen mismatches, but not an MHC1 mismatch, and again, you lose much of the cytotoxicity. You regain this peak. So this told us that the MHC1 is playing a really important role. There may be some subtle variations, something else being recognized, but without doubt, it is conspicuously important for this memory response. So in this case, we did not, to discover what receptor is recognizing it, we did not do a genetic unbiased screen. What we did was we did sort of an intellectual screen where we asked what are the receptors on monocytes that are polymorphic and are able to recognize the MHC molecules. And a small set came up, and one of them were the paired immunoglobulin-like receptors. The reason they're called paired, because they come in a family that contains both activating and inhibitory members. And they have orthologs in humans. They're known as the leukocyte immunoglobulin-like receptors. So in the mouse, the PRAs are stimulatory. The PRB is inhibitory. There are 4 to 6 PRAs, and there is only one PRB in the mouse. In the human, there is a larger number of of the A's, which are stimulatory, and an equal number of the B's, about 6 of each. And the idea here is that they are polymorphic in the sense there are multiple alleles, and they are paired in the sense that one group, the A's, is stimulatory, while the B's are inhibitory. And finally, they bind the MHC class 1 molecules, both in humans as well as in the mouse. So to test whether the peers have anything to do with the monocyte sensing of non-self MHC and the acquisition of the memory phenotype, we studied the memory response to BALB-C, and we blocked the PRA interaction with the MHC class 1 molecule, either by using a fusion protein, by using an antibody, or by generating a PRA knockout mouse where all the alleles of PRA were knocked out in that germline. And in every single case, you can see that we can completely block the memory response, telling us that PRAs are playing a major role in detecting MHC, non-self MHC, and inducing memory. So how do they work? It turns out that the different PRAs have differential interaction with the MHCs. For example, PRA3FC, which I showed you, can beautifully block the response to a BALB/C graft in this case. It doesn't block the response to a C3H graft because C3H carries a different class 1 molecule. We could show in the cytotoxicity assay that PRA3 can block the macrophage cytotoxicity against BALB/C targets, but PRA1, which blocks a different PRA1FC, the fusion protein, doesn't block the cytotoxicity. The cells disappear. We could also show that there's differential binding of the H2DD tetramer, which is the class 1 of the BALB/C, to PRA3 transfected cells. So it binds better than the B tetramer, shown in red here compared to the blue. And we even did additional assays showing that indeed PRAs, the different PRA isotypes, have different affinities for different MHC class 1 molecules. So that endows monocytes with the ability to distinguish between different class 1 molecules based on what PRA they are expressing on their surfaces. So now, before I tell you more about how memory develops, I hope I convinced you it is PRA dependent. It has to do with MHC. Let's first see whether it has any in vivo relevance. And we did that in two models. One is the chronic renal allograft rejection model in the mouse that I mentioned to you earlier. And we did not observe any change in the survival of the grafts when we took away PRA in the knockout, shown here in purple, or we blocked actually, I'll tell you about the blocking in a minute. So when we used the knockout, there was no difference in graft survival, but there was a significant difference in the fibrosis that we were able to detect, as well as the infiltrate. And we analyzed all the cell types that are present. And indeed, we found less T cell infiltration and, of course, much less monocyte-derived dendritic cells. So even though the survival did not change, the chronic fibrosis was reduced. Interestingly, when you take away the inhibitory molecule, the PRB molecule, in a PRB knockout animal, the grafts are rejected quickly now. So this is telling you that the A's and the B's are forming a balance. They're setting the threshold for monocyte activation. And if you take away the brakes, the PRB, then you get accelerated rejection. And I think the most gratifying experiment is that when we took a PRB knockout, and we blocked the PRA interaction, we were able to correct this accelerated rejection and make these grafts last a long time with minimal fibrosis. So this is telling us that at least in a chronic rejection model, these molecules, by modulating the monocyte response, are deciding how long the graft survives and deciding whether you get fibrosis or not. We could do that as well in a heart allograft model, where now we treated the recipient with CTLA-4 Ig or belatacept in humans to allow it to accept grafts for long term. These are fully allogeneic grafts. And you can see here that if we blocked I'm sorry, I apologize. We gave CTLA-4 Ig in a dose that was intermediate. So only a modest number of grafts survived, and they did not survive long-term. They survived only up to about 40 days. But if you combine the CTLA-4 Ig treatment with PRA3FC, now you can get this beautiful long-term survival with absence of manifestations of chronic rejection, such as fibrosis and perivascular infiltrates and intimal thickening in the graft. And if you use PRA3FC alone, it was barely sufficient to prolong survival. It was a modest prolongation in survival. So again, what this is telling us is that these molecules, the PRAs, are modulating monocyte function when you look at the grafts, but alone are not sufficient for rejection, and blocking them alone is not sufficient to block acute rejection. What you can do is you can interfere with the long-term changes that happen in the graft, and when combined with another immunosuppressive agent, you can obtain long-term survival. But again, it opened a new pathway for us, a monocyte pathway, or a new pathway for treatment that doesn't overlap with the agents that we currently use, which target T cells, of course. So what is the mechanism of monocyte memory? How does monocyte memory happen? So this was tough work to do. We did two things. One is the molecular approach, where to look at the transcriptional profiles of the monocytes that are present in either allogeneically or syngeneically stimulated mice. So we took these RAG knockout gamma chain knockout mice, stimulated them with syngeneic or two types of allogenic cells. We isolated the monocytes to high purity, and we did single-cell RNA-seq as well as flow analysis. So let me start with the single-cell RNA-seq, then I'll show you the flow. And to make a long story short, what we discovered is that when you do UMAP and then pseudotime analysis, that these monocytes change their transcriptional profile. They appear to start here, and they end up in multiple places. And it turns out that these multiple places where they end up, the proportions of them there in those endpoints differ whether you are immunizing the mice with syngeneic cells or allogenic cells. So you can see that the allogenic cells favor these endpoints more so than the syngeneic cells. And these endpoints tend to have increased expression of these peer A molecules and reduced expression of the peer B molecule. You can also see that there is what we refer to as variegated expression of the peer A's on the surface of monocytes. We do not have antibodies that can distinguish between the different peer A molecules because they are highly homologous. Instead, we can do by messenger RNA is ask whether in a given monocyte, do you see co-expression of two peer A molecules, or do you see expression of one but not the other? And this is shown in the blue. This shows you that peer A2 is present in a subset of monocytes. Peer A3 is present in another subset, but it's very unlikely that you'll see the two of them together on the same cell. So this is very reminiscent of KIR expression on NK cells, where the variegated expression or the variable expression of receptors for the MHC on different subsets of the NKs or different subpopulations allow them to respond differentially to a non-self while the others don't. And I'll show you that schematically in a minute. But before I do that, I wanted to tell you that by flow, we were able to show that there's clonal expansion of monocytes that is driven by what they're seeing. And I want you to focus only on the middle panel, which is a peer B knockout mouse. And we use tetramers that are either H2D, which is relevant to the bulb C, H2B, which is relevant to the syngeneic B6, or H2K, which is relevant to the third party. And what we found is that if you immunize a mouse with the bulb C, you see preferential expansion of monocytes that recognize the H2D. If you immunize it with C3H, you see preferential expansion in the number as well as the percentages of monocytes that can see the H2K tetramer. And that told us that this is looking now like an antigen-specific response. Or to be more exact, is looking like the NK cell response to MHC that has been shown before. And most gratifying is that when you take away peer A, all these preferential expansions of monocytes goes away. So it tells us that this is a peer A dependent phenomenon. And the reason it was so striking in the peer B knockout is that we took out the interference of peer B with binding, because peer B tends to be promiscuous. It binds to many different MHC molecules. So to summarize, what we think is happening is something similar to the NK cell story, where if you put in a graft with a specific MHC class 1, it will only cause the expansion of those monocytes that happen to express the peer A molecule that recognize that class 1 molecule. And then we went back and asked, what does SERP alpha recognition have to do with it? Because as I told you, we could do an experiment to show that MHC1 disparity is not sufficient, is necessary, but not sufficient for the induction of memory. And I'll skip over this quickly. It turns out that indeed a SERP alpha mismatch and CD47 are required for the induction of memory. So they are required for the primary response, but they're also required for getting the monocytes to the certain threshold where when they now see MHC, non-self MHC, they can become memory. And this is shown schematically here. So there's dual sensing of allogeneic non-self by monocytes. The first pathway has nothing to do with the MHC, has to do with the polymorphic molecule that resides outside the MHC on a separate chromosome. And that is sufficient to drive the primary response, which is the maturation of monocytes into dendritic cells. But if they encounter both a non-self MHC class 1 as well as SERP alpha, and this time they're sensing the MHC class 1 via PRA, then there's expansion of these memory monocytes, or these monocytes that happen to recognize this particular non-self MHC class 1, and those then can generate an even stronger dendritic cell maturation and therefore an immune response. So to summarize, we have all known about Charlie Janeway's and subsequent work by others that the innate immune system is not dumb. It's not a system that simply responds to danger and inflammation, but is a system that can recognize non-self. And that was the discovery of the recognition of microbial pathogens. What I would hope to have shown you today is that this system, the innate system, can also recognize an allograft. It recognizes it with specificity, it has memory, and is capable of clonal expansion, which is totally novel. We had not expected this to happen, but it does happen. So this is not just epigenetic changes. There could be epigenetic changes, but this is good old-fashioned memory. And what are the clinical implications? First, I believe that the innate immune system is perpetually sensing non-self in the allograft, and that's what's leading to chronic rejection, even after danger and inflammation has subsided. Any clinician in the audience knows that even in the most stable patients, if you try to take them off immunosuppression, they will start rejecting. So this could be due to perpetual sensing of non-self, and that innate allorecognition is a potential therapeutic target because it has specificity. You know what the receptors are, so you can target them. And also, as I've shown you in that little clinical snippet, you can even genotype and you can risk stratify patients for the long term. So, of course, the final slide is the most important slide, is to thank many collaborators, mostly Martin Ober-Barnscheid, with whom I've worked for many years, who's an equal partner in this work. He's at the University of Pittsburgh. Jane Danska at the University of Toronto, who really guided us and provided us with congenic mice to genetically map SERP alpha as the cause of the allorecognition. And with Sean Lee at Houston Methodist, who, while we were doing our work, he was doing the macrophage work in parallel, and it all came together, and therefore we decided to publish the work together. And many collaborators here at the University of Pittsburgh and elsewhere who have been essential, and for the NIH funding and the funding from the CERS Endowed Chair. So I'll stop here and see if you have questions, and I'll be happy to answer them.
Thank you so much, Fadi, for this incredible presentation and opening us with new reality, new opportunities, and new potential target. So please, if you have questions, put them in the Q&A section, and I'll be able to read them.
Would you like me to stop sharing, or should I continue just in case?
Maybe continue just in case people want, or maybe, or stop. I don't know if people will need to see your slides again. Waiting for some question. Maybe since there's a lot of clinicians in the room, I see. In terms of, like you talked about the innate immune system perpetually sensing non-self. So in terms of mechanism of chronic rejection, like you say, can you expand a little bit, and how can we use that? And would there be, because normally we think about danger and cell death to monitor the graph and so on, but how could we monitor that and see when it starts again?
Yes. So I mean, that's an excellent question. I think what our work does, it adds a new angle to the pathogenesis of chronic rejection, which is the role of monocytes and the cells that derive from them, dendritic cells and macrophages. Of course, this is not new. There are data in the literature, both experimental as well as clinical, that suggest that the presence of these cells correlate with chronic rejection. What we've done is we've put a molecular face to it, and it's that it's not non-specific inflammation or danger, but it's actually this continuous recognition of non-self, because the graft doesn't go away. And therefore, there are always these monocytes that are playing this trick on us and reawakening the adaptive system, even when we as clinicians thought it was quiet and dormant. So what can you do about it? So one is risk stratification. If you start out by knowing who's mismatched for SERP alpha, for example, that can help. Of course, this will not account for everyone. It will account for only about 20% or so of patients, because the number of patients with SERP alpha mismatches is small. As I mentioned, there's one haplotype that's dominant, especially in the Caucasian populations. And now we're actually trying to advance some PCR methods into the LILRs to know whether we can detect something there. The second is for pathologists, when they do biopsies, is to go beyond the H&E and hopefully do immunophenotyping and tell us, do we see more dendritic cells? Are these dendritic cells in contact with T cells? Are they in the perivascular regions, etc.? So this opens new paths for pathology to inform us on whether chronic rejection is at our doorsteps or not. And finally, and that's the more difficult part, is how would you interrupt these pathways? And I mean, there are some ideas, and we haven't tested any of them yet, and we don't know how much of an effect they have.
No, absolutely. And maybe if we go back, so I see, oh, okay, I see a question here. So maybe just to finish on the pathology, so we do that on biopsies, right? And so you're saying that there will be even, when you take off immunosuppression, you could have this underlying disease or ready to switch. So would you recommend doing regular biopsy or every year biopsy to check for that? Because I have the feeling that you wouldn't sense it necessarily.
I would suggest that, as we had a conversation prior to the lecture, definitely do a one-year biopsy. Because we found data I didn't show that SERP alpha mismatch can predict IFTA, interstitial fibrosis and tubular atrophy, at 1 year. And also there are many studies to show that the one-year biopsy is probably the best correlate with poor long-term outcomes, rather than the 3-month biopsy. The work of Aravind Cherukuri and David Rothstein in Science Translational Medicine lately. Mm-hmm. So I think that a one-year biopsy may be sufficient, because it's early enough to allow you to intervene. And there are ways to, even if you have a one-year biopsy, you can then correlate 3-month biopsy findings or some other measurement with the 1 year, and then even intervene earlier. So that's my vague answer to your really good question.
Thank you.
So the question, I can be happy to read the question in the chat.
Okay, that's perfect. Go ahead.
So that's a really good question. So it goes as follows. It's from Celia. In terms of therapeutic solutions to improve allograft and impair rejection, don't you think that stratifying donor through MHC1 and SERP alpha expression will make it even more difficult to find compatible donors? So I think that, of course, it will make it more difficult. Therefore, what I'm advocating is not to exclude a donor because of the mismatch, but rather know where your recipient stands in terms of risk, and perhaps be either more diligent with your immunosuppression, not to lower it, or if we are clever enough, use some more targeted agents in the future to prevent rejection. So that's how I would answer that first part of the question. Although I can say that in living donors, you may be able to, if there is indeed a SERP alpha mismatch, unlikely you'll find it in a living donor. But if it is, if you had 2 choices of donors, you can choose the one without the mismatch. And finally, in the US, they're doing more and more of these paired exchanges. I'm not in any way advocating that someone gives up a kidney because it's SERP alpha mismatched, but you can imagine these schemes where you have 2 donors, 2 recipients, and you say, well, maybe I should give your kidney to the other person because of this 10% better long-term survival. So the second part of the question is, would it be better to try to target this mismatch with anti-CD47 or even anti-PRA antibodies? So the CD47 is tricky. So as you know, I'm sure you do, the anti-CD47 now is a big hot item in cancer therapy, where the idea there is that you want to unleash the macrophages and the myeloid cells against the tumor because you're not allowing CD47 to deliver the SERP alpha inhibitory signal, right? They do not eat me signal. So because of the way the system works, it works in 2 directions. You would have to come with an agent that specifically blocks the CD47 SERP alpha interaction coming from the donor, but not the one coming from self or from the recipient. So you don't want to get rid of the inhibitory signal, you just want to get rid of the stimulatory signals. And right now we don't have such agents that could really do a great job at distinguishing between the 2. With the anti-PRA, there's a possibility. There are agents out there that are either agonistic or antagonistic to PRB, for example. If you can imagine, if you have an agonist to PRB, you can put on the brakes. And there are people working, there's a Swedish company working on specific anti-PRA or anti-LILRA reagents. So we're a long way from that, but it's possible. And how long do you treat with these antibodies? I have no idea. My guess is that they may be prolonged treatments. But the PRA system doesn't seem like, when we studied the PRA knockout mouse, there are no other immunological abnormalities. So maybe the system is specific and narrow enough that we may be able to use antibodies for extended periods of time without major side effects. The expression of the LILRAs and the PRAs is rather restricted. It's mostly on monocytes, not even on NK cells in some cases, and definitely not on T cells and B cells. And others, there may be some overlap. So these are great questions. I don't think we have the answers yet.
I don't know if you see them. There's more question. One from Nathalie Arbault. Thank you for your very clear presentation. If I understood well, the readout to use to assess monocyte memory is cell number or proliferation of T cells. I would like to know whether you have investigated the transcriptomic of proteomic profile of monocyte upon immunization and immunization plus graft. Do these monocytes change their profile? Do they produce more specific cytokines?
Yeah, I mean, that's a great question. We have a fellow now, Jason Ossar, who's working on this. And what he's doing is he's very carefully dissecting the transcriptional as well as the epigenetic profile of the monocytes, both over time, as well as a comparison between a PRA knockout and a wild type, to ask if there is a specific memory state in these cells. So is it just that the monocytes increase their number and that's why we see memory? Or is it that there are qualitative changes in the monocytes themselves, whether at the transcriptional or at the epigenetic level, which of course are tied together. That's why we do them together. So we don't have an answer yet. We'd like to think that there are qualitative changes. There is a memory monocyte, like you can say there's a memory T cell, and it's not just a matter of increased numbers of these cells. So I hope I answered the question. If not, feel free to repeat. But that's really it's a great question, and it's a 5-year project. I hope we'll finish in 3 years. I'm getting tired.
Maybe one last question. We have a couple. Maybe you can answer them by typing after. But Nicolas Chaumont, fascinating presentation. I think you covered the first question, possible to block CD47 in humans to prevent graft, and then also SERP alpha. But the question too was, what is the extent of monocyte proliferation in terms of number of cell generated? Is it comparable to T cells?
No, it's not. it is not. In some experiments, it's been very variable, and I have to confess this is one of the Achilles heels of our study. So in some cases, we observed like the spleen gets bigger after you immunize the mouse, and we could see up to a 5-fold increase in the number of monocytes. But it's nothing compared to T cells where you can get 1,000-fold increase in number. We'd like to do it in a more specific way by looking at the MHC tetramer positive monocytes, but we haven't done that yet. And same with BRDU uptake or EDU uptake. You can see small percentages, but you don't. It's T cells are the best. Monocytes are more primitive.
Excellent. If there's more questions, but for the sake of time, I think we need to stop now, which is unfortunate. Next time you come here, Fadi, and we take time for longer for questions.
Melanie, I'd be happy to stay on because I'm fine. If you wish to, it's up to the audience and you.
Okay, so maybe Marie-Chantal, your call.
Yes, thank you very much, Fadi, for your presentation. Alors, tout le monde, je veux juste vous rappeler que le prochain séminaire de transplantation, ça va être le 22 octobre prochain. Puis, on va avoir la chance d'avoir Edith qui va nous faire, Edith Renaud, qui va nous faire une mise à jour sur le BK. Donc, moi, je vais devoir, I will have to leave, Dr. Lakkis, so thank you very much for your presentation. Alors, je vous dis à tout le monde, on se voit le 22 octobre. Au revoir.
Merci, Marie-Chantal. Fadi, we can stay for a few minutes to answer the last two questions if you want.
Sure. Yeah,
for people that want to stay, if you want to leave, you can leave. But people who want to ask the question, let's take this opportunity. So Monique Bernard asked, what would be the implication for cancer?
Yes. So I don't know of any implications of the peer system for cancer. I did mention the CD47 SERP alpha. So there are really good studies showing that if you block the SERP alpha signal or block CD47 from interacting with SERP alpha, you could enhance the clearance of tumors such as non-Hodgkin's lymphoma, I believe, either Hodgkin's or non-Hodgkin's. It was a very nice New England paper. So if you were to apply this for transplantation, you have to be careful not to be blocking the inhibitory signal. You just want to block the stimulatory CD47 signal. But if I were to use my imagination, I really think that cancer immunologists should look in this area the way we did in transplantation and move away from non-specific danger or inflammatory signals that they think drive the dendritic cells in cancer. If you have a tumor with a high mutational burden, for all practical purposes, this is like a multiple minor antigen mismatch, right? Just like putting a bone marrow transplant that gets rejected or causes graft versus host disease. So if we were to understand what drives a dendritic cell maturation in a tumor and go beyond inflammation or DNA or RNA, but get into whether there is some recognition of non-self that's happening. I'm not saying that it's going to be MHC based, so maybe the peers may have no role there, but just thinking along those lines maybe would uncover more checkpoints that cancer therapists can use, but this is entirely speculation. So think like transplanters for a change.
Exactly. Well, thank you so much for joining us today, Fadi. Like I said, next time in person, face to face. So thank you for this excellent presentation, and hopefully we'll have you soon here in Montreal.
Thank you, Melanie, and hopefully we'll see everyone at the big meetings. And as always, it was a great pleasure.
Thank you so much. Have a great day.
You too. Bye-bye.
Bye.